Polymerase chain reaction is a technique that scientists use in order to copy a lot of DNA. So you would start with one small piece of DNA, maybe it's from a crime scene, for example, and you can't really do much with one piece of DNA. You can't really sequence it. You can't really do a DNA fingerprint with it. We'll talk about both of those techniques later, by the way, but you can't do much with this because you don't have very much of it. So you're going to use a polymerase chain reaction in order to get a lot of it. So a polymerase chain reaction can start with just a small sample of DNA and produce lots and lots and lots and lots more DNA. So a polymerase chain reaction can amplify the DNA. That means produce many copies of it. And that's of a specific target sequence of DNA. So here's how it works. If you think of the natural way that we produce more DNA, what do we need? Well, we need helicase, right, to unwind the DNA. And then we need primase to make a short RNA primer. And let's see, we need single-stranded binding proteins. And we need DNA polymerase. So this technique doesn't use all of this, all of these materials, but it does need to do all of the same jobs that these materials do. I think I got nucleotides. I got to put that in here, nucleotides. So this PCR doesn't use all of this stuff, but it does do the same job as the things that these do. So here we go. We start with a single small strand of DNA, and we want to produce lots and lots and lots of copies. So we've got nucleotides that we need. We actually don't use primase to make a primer. We buy the primers from a company or make them in the lab. So here's the primer. So we don't actually use the enzyme primase in this reaction. We make them ahead of time or we buy them. So for example, if I want to do a lab at the school, in the biotechnology class, we do a lab on GMOs, so genetically modified organisms, and we just buy the primer from a company, and that's a small segment of RNA, and it's going to be complementary to, in the case of this lab, that sometimes in the biotechnology class, this primer is complementary to a small segment that's found in most genetically modified plants. So anyway, you've got your primer, but you don't need the primase. Instead of helicase, the job of helicase is to unwind the DNA. We actually don't use helicase to do that. Instead, we use heat. So we have a machine. We actually have a machine in the biotechnology room called a thermal cycler, which is amazing, amazing that we have at the high school level. But anyway, what it does is it heats up a sample and then it cools it down. And it does that over and over and over again. Each time it heats and then cools, you have one complete cycle of what I'm going to show you here. And then it would heat it up again and do it all over again. So anyway, the first thing that it does is heat up the DNA so that it separates out. And then the primer can attach, if this is an A, it would match a T. If that's a C, it would match a G, right? So this RNA primer will match the DNA that you're interested in replicating. And DNA polymerase is going to come along and match these nucleotides into where they go here, and you've just copied DNA. Instead of one molecule of DNA, now you have two molecules of DNA. And the thermal cycler is this machine that will then heat it all up again, right? We don't actually have the single-stranded binding proteins. This is usually done on small pieces of DNA, and the heat typically, when it opens up, the RNA primer matches in, so it usually doesn't go back together. But we do need the nucleotides and we do need the DNA polymerase. So the requirements are DNA polymerase, DNA primers that match the sequence that you're interested in, and then this machine that will heat and cool the sample for you. So one thing that you might remember about DNA polymerase, ACE, is that it's an enzyme. And most enzymes are proteins. And what happens to proteins when they get hot? They denature. So the whole key to this PCR technique is that scientists use a DNA polymerase that they isolated from a bacterium that lives in hot springs. So it can actually stand very, very, very hot temperatures. How cool is that? So the key, oh, sorry, it says that here. The key to PCR is this unusual heat-stable DNA polymerase called Taq polymerase. It's named after thermophilic bacterium Thermus aquaticus, which was originally isolated in 1976. I was just a little kid then.